Hello, my dear class. I am recording a lecture for you today, which is kind of the broad brush picture of systematics and phylogenetics. And so my goals are to kind of teach you about the broad structure of some big subfields, essentially, of evolutionary biology, and end with the phylogeny of the angiosperms, which is what we're kind of going to be exploring in chunks through our class. There are some handouts in your packet about this, and hopefully it'll be clear which ones to look at as we go. Okay, so terms like systematics, taxonomy, let's kind of unpack some of this idea. Well, systematics is kind of the broadest of the possible terms that we're using here. And it's all about the structure of biodiversity. And subfields within that are taxonomy, that we're going to talk quite a bit about, phylogenetics, which we'll cover briefly, and biosystematics, which we'll cover briefly as well. I would say that if you find these things interesting, my biogeography and phylogenetics course, which I'm teaching next block 5, goes into these last two in a lot of detail. Okay, so let's start with taxonomy. So taxonomy is all about how to identify something. What is it named? The process of nomenclature, which I will do a whole lecture on that for you. Classification, the ordering and the grouping of things. And certainly the need to have consistent names and consistent groupings that are predictive and meaningful has been with us for many, many centuries. Okay. So here is an example species, Cirsium pitcherii, described by Eaton. Here's a picture of Eaton. And this is a member of the family Asteraceae that we're going to talk about. And the common name is dune thistle. Okay, and it is endemic only to the Grand Sable Dunes region of Michigan. So on the west side of Lake Michigan, you have these beautiful dune systems. And this is where this really beautiful thistle is found. Now, speaking of thistles, our dear friend Jen Ackerfeld, your textbook author, who we'll get to see, this group, the thistle group, is what she did her entire PhD on. So she is currently the world's expert on this group. Thistles are great. They're in the royal symbol of the royal family of England. Fun little fact on that. Okay, so the original sample, one plant that was collected from this was found here at Grand Sable Dunes. So that's a type specimen. We'll talk about the process of creating types and specimens in a subsequent lecture. Okay, so it has a name, it was described by someone, and it's backed up by a specimen. Okay, so phylogenetics would then say, to what is this dune thistle related? Who are its relatives? What is the relative order of common ancestry that it shares with other things that are alive today? And it turns out that this much meatier, chunkier friend, Cirsium canescens, the prairie thistle, has been determined to be the closest relative of the dune thistle. And you can kind of see the close resemblance between them, even without knowing kind of the technical terms. So you would say they are sister species, the two closest relatives depicted in a phylogeny. A phylogeny is a branching, often bifurcating depiction of evolutionary history. Now, caution, when we go keying something out, right, that is a bifurcating document, the either or choices, et cetera, et cetera. In almost all cases, the key to species is not reflective of phylogeny. It can be that the closest relatives end up fairly close to each other in a key, but it's not necessarily so. I go into a lot of this in other classes, but there's very specific characters morphologically speaking, and increasingly more so DNA that are used to build or to uncover the true phylogeny. Many of the features that we see with our eyes are shaped by convergent evolution and determining their homology, determining that they evolved once in a common ancestor as opposed to independently, is a really tricky thing to do. And so increasingly, phylogenies are based more and more on DNA, which is a much more objective a series of traits that vary than morphology. Okay. I don't know if any of that makes sense. Ask me questions in class if not. Okay, so there once was a creature that is no longer alive today. That creature was the common ancestor of both the dune and the prairie thistles. But of course, there are a series of nodes. Yep, same term that we do with morphology, right? The common ancestors are those branching points to the currently alive species today. So if we go back in time, there was an ancestor of all thistles, all members of the genus Cirsium, which all share a single common ancestor. Because they all share a single common ancestor, Cirsium is monophyletic. However, our dear friend Jen Ackerfeld would probably critique everything I just said because her PhD work on using DNA, looking broadly across the Cirsiums, actually finds a lot of complexity within it. And in fact, it is not monophyletic, and she's actually had to author some new genera, which is very exciting. Maybe we'll get her to talk a little bit about that. Okay, and then here is something that is not in the genus Cirsium for our example here, right? And this is the genus Carduus. So similar from what you're looking at with your eyes, right? So it would share an older common ancestor with the genus Cirsium. So the relative recency of those three common ancestors shown in blue determines relative relatedness. The more recent an ancestor was alive, the more closely related its descendants are. So in this example, the common ancestor of Cirsium canescens and Cirsium pitcherrii lived more recently than the other two. Therefore, those two are sister, okay? Those two also share the other two common ancestors as well, but they're more distant in time. So we're really only focusing on the most common recent ancestor of whatever we're comparing to determine relative relatedness. That is the process of phylogenetics. And one of the major goals of systematics is to place all living things within a grand phylogeny of life. And so that is what I do professionally, is I am trying to build the grand definitive phylogenies of the couple groups that I'm a systematics expert on. So the genus Puya in the Bromeliaceae plant family, and the entirety of the plant family Goodeniaceae, if you were interested in knowing that. Then biosystematics asks all kinds of exciting why questions. So how, when, and where did this speciation occur? So there was an ancestral lineage and it split into two. Why did it do that? Where did this happen? When, geologically speaking? And what caused it? What caused one thing to split into two? So you might consider like the distribution of the species today. And then you might want to know the time period in which that common ancestor in blue was alive. So in this case, it's really important to know things like there once was a massive ice sheet covering much of the upper Midwest, including the entirety of Michigan, and then it retreated. And the expansion and contraction of these various ice sheets caused various allopatric speciation barriers, kind of segregating different potential populations of that ancestor for thousands of years. Then when the glaciers retreated, enough time might have passed that they were now independent species. Okay. So just giving you a taste of some of the fun questions that you can ask within the broad field of systematics. So, some of the goals of systematics are to inventory Earth's biota. And we've done, a pretty good job so far at describing and discovering new species. But obviously, there is a ton of work to do in new species discovery. I have been adjacent to new species of angiosperms being discovered. I thought I had one on my own once. It turns out not to have been a new species, just a new record of an existing species from a part of Peru that no one knew it was there. So that was the closest I independently got to that. But I work with botanists all the time that describe new species. Jen Ackerfeld describes new species, which is very cool. Certainly, it's the case, though, that you're not equally likely to find a new monkey species as you are to find a new fungus. Angiosperms are in pretty good shape compared to all of this. Certainly, insects and microbial diversity are the most problematic of all of the life forms. Now, where on Earth does this work have to happen? Well, in temperate latitudes, right? Including where we're at in Colorado, the inventory is nearly complete. It's not really the case that you're discovering lots of new species in Colorado in angiosperms regularly. Most of that work happened a long time ago. With new DNA evidence, it can present itself that there's cryptic species or we have to redefine species boundaries. But it's not like we're going to a new valley that has never been inventoried by Western science before and finding a bunch of stuff. That said, there are still some really exciting things. So it was not even 20 years ago, I guess now. But in Alabama, there were a group of botanists going on a river trip, and they saw these bluffs up above the river. And they went up there and they described, I think it was like 10 or 15 new species of angiosperms, like big, obvious families, whole new species that had never been described before from these bluffs. And that was really cool to see. And actually, Alabama is the third most biodiverse state in the contiguous United States. So there is still sometimes cool things to do, but for the most part, the temperate inventory is nearly complete. Now, there is still interesting academic work to do on regional or local floras. There are botanists who produce kind of definitive, or at least for a period of time, definitive work describing all the things there. So my predecessor, Tass Kelso, did a beautiful kind of floristic book about the flora of the Pikes Peak region, which we do have access to here as a class. The big one from Botany is the Flora of North America series of books. We have all of the books in our classroom, which you can look at, which is the definitive treatment or taxonomic description of all of the major groups of plants here in North America. And they're producing a couple volumes of this multi-volume book series every couple years, and it's pretty darn cool. Now, of course, there's a ton of work still to be done in the tropics, and tropical systematics is still at that cool species discovery, not even really having a sense of the magnitude of diversity in really cool places. Like this is the Venezuela Guyana shown here. Okay. So then once we have discovered things, we want to be able to produce keys, like the flora of Colorado, be able to identify and put names on them. That's the process of taxonomy. We'll talk about this a ton more, but for now, of course, right, we start with the species name, the Latin binomial like Solidago canadensis. You all know this, genus plus specific epithet equals the species name. Common name in English would be the Canada goldenrod. And there is a lot more we'll talk about related to that. Beyond that, though, we want to situate this species within a broader taxonomic framework that's logical, that makes sense, grouping organisms based on relatedness, hopefully with features that make sense and that are readily discernible by lots of scientists that use this kind of information. So essentially, we need to create an encyclopedia to organize the vocabulary of names. This is all very relevant, especially when we have specimens that are placed in an herbarium or a press-dried plant collection. There must also be a specimen tied to every single name. And so we want that to all be very organized in a way that makes evolutionary sense. Right, so again, we've talked about this, you've seen this, right? But here is example of the Linnaean hierarchical classification system for that Canadian goldenrod, Solidago canadensis. The Latin of genus and specific epithet have to agree, so that's part of this. Specific to the botanical community, you cannot have the genus and the specific epithet be the same. I just saw in Heather's beautiful OB Day presentation that in animals, they can be the same. So that's one way in which the plant and animal name systems can differ. You would never see Solidago, Solidago in plants, but what was it? Gulo gulo, that is a species of animal. Sometimes there's tribes and things kind of below the family level, subfamilies. Here's the Latinized endings. So tribe would be EA, so Asterie, the subfamily Asteroidea. And then the ones we've been learning certainly are families, ACE, so Asteraceae. Above that are orders. You will see the names of the orders of angiosperms in the phylogeny I'm going to share with you. So Asteraceae is within the Asterales. And then both the class and the phylum, then we switch to the Magnoliopsida and Magnoliophyta, okay? So that is getting to the whole angiosperms groups up there. I don't personally think the classes are very useful, but orders and family are the kind of things that I think for serious botanists that are kind of useful to just kind of rote memorize some of them. Okay, which brings me to the final thing I wanted to show you, which is the phylogeny of angiosperms. I am unabashed in this class at making this an angiosperm class, though I want you all to be, continually aware of the broader phylogeny of land plants, right? So reminding yourself that we have mosses, hornworts, and liverworts as the earliest diverging land plants. There's a wee phylogeny in your packet about this. Then we have ferns and fern allies as the earliest diverging vascular plants with xylem and phloem, right? Then we have the gymnosperms, our seed plants that lack a fruit. And then this is the angiosperms. And we have that monster poster in the lab that you can look at if you like geeking out about this. So there is a long and very nerdy history of modification. So the original discovery of this phylogeny and modifications of it. People have been describing groups of plants going to Linnaeus and actually prior to him. This got formalized over and over and over again, changed, moved, whatever. But starting in the late 90s, there was the first definitive phylogeny of all angiosperms based on molecular sequence data. So the era of fairly quick and cheap gene sequencing with which to build phylogenies with strong statistical support dates to the 90s. And there's a group of botanists globally called the Angiosperm Phylogeny group of people. They and their labs produced these various phylogenies across the time with more and better data. So we are now on the fourth iteration of the APG, as it's called. So what you're seeing here is the APG4, which was published in 2016. And I mean, my PhD advisor is involved with this, so I am ancillarily related to all of this. Okay. So I want to kind of situate the groups of plants that we're emphasizing in this class within this structure. So there are certain things that we straight up don't see at all or very rarely in Colorado, and then certain groups that are very, very common. So that's kind of how I've tried to emphasize what I'm going to teach to you. But here's the whole picture. And I've emphasized here, highlighted in bright blue, various groups I'd like to become moderately familiar with. In the rainbow colors, I'm highlighting the chunks of orders-ish that we're going to learn kind of as sets. Okay, so now I'm going to talk through this. Angiosperm itself, right, is a monophyletic clade, meaning that flowering plants evolved once. All the flowering plants that we see today share a single common ancestor. Okay, so I'm starting from the bottom of this diagram and kind of talking my way up. So there is a single species placed in the Amberolales, that's on the left, kind of oriented vertically. It is a very modest shrub on a rare island, or a obscure island called New Caledonia, which is in the South Pacific. And it alone is sistered to the rest of the angiosperms, meaning it alone is equally closely related to all other flowering plants. That's wild, right? That's so crazy. Okay, then some of these other groups kind of on the left, all over the globe, but things like the nymphaeales, that's including the water lilies. So we've talked about water lily a bit in class. And then this magnolids clade includes things like magnolias, black pepper, bay laurel, lots of trees with lots of aromatic compounds, mostly species rich in much moister places than here. So if I was teaching you this class in the eastern half of the US or the southern part of the US, we would have examples of these to show, but we essentially have none of these here in Colorado. Okay, so then we have the deep split, back to kind of reading the horizontally oriented words, between the monocots and the eudicots. So you might have learned monocots versus dicots. It's fine for intro, but for me, I'd like you to learn the nerdy word eudicots. The reason why eudicot would mean true dicot. You cannot split all of the flowering plants into monocots and dicots because the magnolids and the stuff to the left of that don't fit into either of those. So hence why eudicot is true dicot. Okay. At any rate, monocots, you all remember this kind of stuff, parts of in multiples of 3, parallel venation, fibrous roots, lacking a taproot, etc., etc. That group has been on its own, doing its own thing evolutionarily for a really long time. And it's quite different in many respects from the eudicots. The first group of plants that we are going to learn is the ones in orange. And I will do you a lecture on those next. So I'm calling those the non-graminoid monocots, meaning the non-grassy-like monocots. The graminoids are the little friend in pink, the poales that we'll teach at the very end because they're really challenging. But those are the two main groups of monocots. There's lots of really important tropical groups in there, as well as freshwater aquatic flowering plants that we'll talk about briefly, as well as things like the palms in there. Okay, over into the eudicots then, you see highlighted in yellow what I call a basal grade of various orders. So they do not form a clade, they form a grade. And these tend to have lots of parts per whorl lacking a lot of fusion. So this is the basal eudicots that we'll be learning soon too. We will be talking about friends like the ribes, the currant that we've talked about, columbines, things like this are in that group. Okay. Then you can see, tracking to the right, we have two deep splits in the phylogeny of the Dicots after that. Oh, backing up, Dicots do share features of pollen, having 3 pores per pollen grain, as well as some other features. Again, kind of that baseline multipliers of 4s or 5s for the parts. Now, of course, there are going to be a few exceptions to that that can be kind of tricky. Okay, so then the remainder of the Eudicot splits into 2 major clades, the superrosids and the superasterids. Rosids to the left are in general going to have free parts, like not a lot of fusion, particularly between petals. The asterids, in contrast, are going to have a lot of fusion, and have a reduced number of things like stamens and pistils. There's going to be exceptions, but that's kind of a general take home there. Okay, so rosids we're going to split into 2 subclades, the 2 colors of green, the fabids and the malvids. I will teach you those groups. Fabids are going to include bees, peas and beans, Rosaceae, the Salicaceae, some things we've seen. Malvids are going to include the Brassicaceae we've seen, and a lot of our trees as well. So those are the 2 groups we'll cover there. Then within the superasterids, there's another grade of different orders. Again, not sharing a single feature, but kind of one-offs along in that light blue color there. I call that group the superasterids. That's where we'll find cactus, and a lot of, I don't know, shrubs, flowers, all kinds of things there. And then we move into the true asterids group, which there are 2 major clades, the campanulids and the lamids. And that's kind of where we will wrap up the class, particularly within the Asterales, the Asteraceae, and the campanulid clade. Okay, I hope that is somewhat useful. I find phylogenies very useful as opposed to just learning like disembodied plant groups alphabetically or something like that. Finally, there is this figure here, which is the exact same phylogeny, but highly formatted with a lot of information. So it's, turned 90 degrees from the way the other one was. All those same clades are labeled. There is text information about features shared within that clade on the branches. If you like to nerd out about this stuff, if you really like to nerd out about this stuff, I want to know, because this is like what my research is. I do include chunks of this in your packet, so hopefully you can read some of that text. This is also available online. I will put a link to this file. To the right, in the colored bands, are the families within those orders. And in my lectures about groups of plants, I'm going to try to highlight which are the families that are relevant here. So I hope that's a useful way to kind of understand this and understand how I'm going to use phylogenies in this class to arrange and orient some information. And let me know if you have any questions. Thanks, guys.